Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Getty Hamblin, Field Application Scientist from Mirus Bio.
Hello everyone, my name is Getty Hamblin, Field Application Scientist with Mirus Bio. Today we will be discussing bioprocess considerations for large-scale transfection using transit virus gen. We'll kick off the webinar with a bit of background on Mirus and virus gen transfection reagent. We will then move into some of the key considerations and challenges around transfection, particularly at large scale. We will then highlight the unique features of virus gen and how they overcome these challenges. Finally, we will wrap up and open the floor to any questions you may have. A little about Mirus. Based in Madison with a worldwide presence and founded in 1995, Mirus employs a team of biologists and chemists to formulate novel and optimal formulations for transfection, specific to cell types and applications. Mirus has joined under the Gamma Biosciences umbrella and joins companies such as Astreo Bioseparations, Univercells Technologies, NanoParel, Biomagnetic Solutions, and Nirin to enable innovation and provide end-to-end solutions for cell and gene therapies, vaccines, and biologics. Now, let's dive deeper into transit virus gen. Transit virus gen gives more yield than competitor reagents, leading to more process savings, supported by more expertise of R&D and application scientists. Virus gen is our reagent specific for production of lenti and AAV. Available as RUO to full GMP, virus gen is fully synthetic, chemically defined, and has no licensing requirements. Customers can use virus gen standalone in their viral vector manufacturing or combined with a complex formation solution and enhancer specific to the virus that they're making, be that AAV or lentivirus. These enhancers are likewise available in RUO or GMP configurations, fully chemically defined and animal origin free. To support our GMP customers, we have developed a residual reagent and identity assay for virus gen, the drug master file finishing early next year. We have likewise launched a residual reagent and identity assay for our complex formation and enhancer solutions for both the AAV and lentivirus kits. Virus gen, like any GMP reagent, has been tested for lot-to-lot comparability via both physical titers, these would be DNA-based assays such as genome copies, as well as functional or infectivity assays measured in transducing units per mil. Virus gen comes in various grades and volumes to support you in every step of bioproduction. All grades are chemically identical, differing only on testing, packaging, and manufacturing quality. Virus gen yields higher AAV titers than competitors standing alone, and by logarithmic orders using the enhancer. For lentivirus, titers are 2 to 9 times higher, either using virus gen or virus gen plus the enhancer, respectively. Transfection might only be one small step in a viral vector process, but it is one of the key steps in getting higher titers. Higher titers means fewer runs, which means less labor, less facility capex, and ultimately means that you can get more virus for less cost, giving you more savings in your process. And now we'll look at some of those key considerations for transfection workflows that are important to getting those higher titers. This is a standard workflow for virus gen, be it for an AAV or lentivirus manufacturing process. This process can work from a well plate to 1,000-liter plus manufacturing. Of note, we highlight some of the key parameters around transfection: cell density, cell viability, complex formation time and methodology, and ideal harvest time. We will need to keep these factors in the back of our head as we develop and scale our viral vector process. So here we dive deeper into some of those transfection workflow steps. So early on, we're going to be considering all of these, but once we get into more of our bioproduction steps, we've fairly well optimized the cell density, the reagent DNA ratios, and harvest time, and these will be somewhat locked into our process. So going to that complex formation step, this is where we're going to be looking at really some of those critical parameters that are both the biggest challenges in scaling as well as some of the greatest things VirusGen is able to overcome in this step. So looking at the complex formation itself, this is fairly easily done through a transfection worksheet that Mirus will provide all customers. It is simple to follow formula, and this can be used at, again, the small scale or quickly scaled up. We have some customers that have used this worksheet directly into their batch records for complex formation. Of example, we follow this formula to do a few permutations, either being a 2 ml culture in a 6 well plate, a 30 ml culture in a 125 ml shake flask, or directly to a 150 liter culture in a 200 liter bioreactor. Now, specifically going to the complex formation step, let's take a deeper dive here. And for this section, I'd like you to imagine yourself in your own viral vector process. It is the day of transfection, and you're about to go about forming your complex. So when we form complexes, one of the first questions is how much complex to make? What will be the total volume of complex? Mirus suggests for transfection that we would form the complex based on the total volume of culture, and that we would use 10% of that volume. So say we have 150 liters in our bioreactor. We would need to form a 15-liter complex. We can refer back to the sheet we provided earlier to get the numbers of exactly how much media, DNA, and reagent we would be using. And the volume is pretty critical when we scale up, because the larger volumes will require larger equipment, processing time, mixing time. So just take, for example, we were trying to scale a process that we developed, it had an ideal transfection, complex volume of 25% of total reactor volume. If our process was 2 liters, that's not going to be too hard to form for a 250 ml complex. But say we scaled up to 1,000 liters. Now we're looking at 250 liters of complex, and this could really be a major logistical hurdle to move and manipulate that much liquid in a very critical and tight time frame. So just something to keep in mind when we're looking at the upper end. And a lot of times we might be thinking, well, what about the other option? Could we make our complex more concentrated? Could we use a smaller volume? So a more concentrated volume is going to concentrate the DNA and concentrate the reagent, both of which might form complexes faster. They're going to find each other in the media to actually make that complex more quickly, and they're going to be more concentrated. So we know that a transfection process is not exactly gentle on the cells, and that when you do this, you are going to be stressing your cells. And the good news here, and if we look at the graph, is that regardless of if you use a 10% or 5% using different multiple cell lines, or versus PBS, you're going to get a better yield regardless of if you use a 10 or 5% concentration. And we'll look at kind of things to optimize as far as that formation of volume in the next couple of slides. So for a viral vector process, our complex tends to be 1 to 4 plasmids, VirusGen, and our formation solution. That can be our media, phosphate buffered saline, or our complex formation solution. And as we get those individual components into one bag or mixing operation to make that complex, we're going to probably need to filter sterilize one or more of these components. And the good news here is VirusGen can be filter sterilized multiple times without any issue. So this allows for flexibility in your buffer prep, or for a non-closed system to go into a closed system, or even for logistical ease when you have operations across different scales and areas in our manufacturing space. You might be preparing this in a buffer prep area, moving into the manufacturing floor, or need to filter directly in the manufacturing area. We do need to keep in mind, though, that once our reagent is going to form a complex, And once our reagent finds DNA, it will start to complex it. And those particles will start to increase in size, growing larger over time. And that while the individual components can be filter sterilized into the complexation vessel without issue, once they've met each other, we are now in a closed system. So keeping in mind that our initial formation can be an open to close via filter sterilization, from the point that we start the complexation, we are going to be in a closed system. So this means working with welding, aseptic connectors, and things of that nature. So as we noted before, once the reagent and DNA find each other, they're going to start to complex. And as this happens, we are going to have a window of time for optimal transfection. And for VirusGen, that window is a very open, wide time frame of 15 to 60 minutes. So this allows for a lot of flexibility in where our complexes form. So again, we could form this in a buffer prep area versus the dedicated upstream suite. The time to make and verify those aseptic connections. Welding can take a few minutes per line, verifying the connections. And of course, we want to ensure proper documentation across the process. So we're not trying to rush this step and hurry our operators and giving them ample time to properly perform this step. Likewise, VirusGen is stable at room temperature over time and actually performs slightly better at room temperature versus a chilled system. So this would eliminate any need for jacketed systems or measuring or recording temperature of that complex as a critical process parameter. You can simply make this and have it as a room temperature feed, eliminating a lot of the logistical and utility headaches around cooled feeds. And so throughout that complex formation window, the complex will be stable. It is worth noting that most processes, especially AAVs, depending on serotype, cell line, will tend to have an optimal time. Your process development group has probably determined a sweet spot. It might be the 30 minutes. It might be 30 to 45 minutes. It might be closer to the smaller volume in a 10 to 15. But once you're within that, you're not going to have a major risk or failure. The complex won't fall apart and will be stable throughout that time frame, giving again your process very flexibility, a lot of openness for your operators to correctly perform this step. One other thing that's very nice is that VirusGen is best formed in static conditions. And that means that you don't have to have a dedicated mixing system. You don't have to have agitation, rocking dedicated during any amount of time. After an initial homogenization of the individual components of the complex, you would then be ready to just leave the complex by itself. And this would sit for the time that you've determined and then be ready to go. So again, you don't have to have a dedicated equipment or unit operation to mixing. So now let's bring it all together and show a real workflow for a VirusGen process at the 100-liter scale. So this is from an actual customer process. And here we see that the customer would then filter the DNA and complex formation into a 2D style mixing bag. This could even be a wave style bioreactor bag. And then they would filter sterilize that VirusGen directly into that mixture. Here, that's just a quick mix, around 5 seconds, not even a specific point. And then a 15-minute incubation before being welded onto a stirred tank reactor and either being gravity drained or pumped into the bioreactor. Of note, and a question we do get from time to time, is pumping versus gravity draining. And as long as your pump isn't aggressively pumping at high speeds or high pressures, the shear force that the complex will see for most pumping is not going to be too harsh to damage the complex. Certainly figure out what is best for your process, what works for you, and either one will be able to be used with VirusGen. So kind of looking at that as a deeper dive, here's some of the notes on the back end. So in this case, the process uses about 5 liters of the complex formation solution and enhancer and 300 milligrams of DNA. This is optimized for 1.5 milligrams DNA per liter, 3 to 1.5 ratio to total DNA. Again, things you will have optimized in the early step. Then in the second bag, you'd be doing the filter sterilization of the complex formation solution enhancer into the bioprocess bag, and then also filter sterilizing the VirusGen reagent into the filter rinsing and then doing a second rinse with complex formation solution enhancer, mixing well. Next, using a wave style bag, here giving it a quick gentle mix, only a 12-degree angle, a 25-degree C temperature with just 5 RPMs, just holding it at room temp. After letting that sit for 15 minutes, in this point, they would then weld that complex onto the bioreactor and then gravity drain it. Now, what we described was somewhat more the optimal conditions, or something that we would say would be used that would best align with the stock VirusGen protocol. But there are some slight differences that you could do, is where instead of mixing the DNA and complex formation solution and then filtering the complex formation solution into the bag, you could either do that separately or all together. So this might be something you're familiar with at small scale, forming it as a 1-tube or 2-tube complex. And it doesn't really matter which one. Both of these have been used successfully at that 200-liter scale to successfully transfect a large bioreactor. So regardless of how you want to form that complex, VirusGen is very open to whatever works best for your process. So now let's summarize and go over some of the resources you have. So when you get into transient transfection at large scale, there are significant challenges that really come with the scale that are not just the standard challenges we see in a viral vector process. And VirusGen brings a lot of unique advantages that bring, something different at large scale that will not only bring you higher titers, but it's just a better process all around. And transient VirusGen GMP reagents will offer more yield than the competitor, delivering the DNA better to your Hex-293 cells for higher quality AAVs and lentis, which of course will lead to more savings. Higher titers with higher full empty capsid ratios provide more usable virus per run, and that's all backed by more expertise. So you have the full backing of Miras's scientific team to accelerate that journey from R&D to GMP. And if you'd like to reach out, please contact us for an evaluation of VirusGen for your process. And at this point, I'd like to thank you very much for your attention and open the floor to any questions you may have.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what is the largest scale bioreactor VirusGen has been used with in a successful viral vector process?
So the largest customers we have currently using VirusGen go up to about 1,000-liter scale, with some proposed work as far as 2,000 liters.
And is VirusGen compatible with non-stirred tank bioreactors, example fixed bed bioreactors?
Yes, so we have several customers using our reagent in an example, say an iCELLis style bioreactor. We have also partnered through Gamma with Universal Cells Technologies using their hydrocarbo and nanoscale bioreactors that are also fixed bed bioreactors.
Okay, great. So it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Getty, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is a media change required with VirusGen pre or post-transfection?
No. Like many Miras reagents, we do not suggest performing a media change pre or post-transfection. You might need to dilute your cells down to a specific cell density, but after adding VirusGen, you do not need to do a media change.
Actually, it looks like we just got one more question coming in that we could ask. It says, what is the highest VCD we do a transfection with the reagent?
So our standard suggestion for VCD would be between 2 and 4 million cells per mil for a standard viral vector process using Hex cells. Depending on your process, that number might be slightly lower or slightly higher. But our standard suggestion would be the 2 to 4 million range. So to answer would be 4 million cells per mil.
Okay, great. Well, thank you, Getty. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.